Good morning, everyone. Thank you for joining us in this webinar on bacterial canker of tomatoes. My name is Jaime Piñero, and I work for Lincoln University, Cooperative Extension. This webinar has been offered in response to outbreaks of bacterial canker that have been documented in various states in the Midwest, for example, in Northwest Missouri, 2011, 2012, but also in Ohio. So this is how we are going to proceed. For the first 30 minutes, 40 minutes, Dr. Dan Egel from Purdue University, he's going to be presenting on how to diagnose bacterial canker in the greenhouse and in the field. And then he will continue describing the disease development and spread routes. Good morning, Dan. Can you hear me?
Good morning. I'm here.
Thank you. And after Dan Egel, Dr. Sally Miller, she works for the Ohio State University. She's going to present on disease prevention practices and also management options. So feel free to ask questions using the chat box. And then we have about 90 minutes for this webinar. So I will stop sharing this file now. And the first speaker is Dr. Dan Egel. Go ahead, Dan.
Good morning. I'm going to go ahead and I'm going to watch the chat box. If people can't hear me or have problems, then please let me know.
I can hear him fine.
All right. What I'd like to do first is present an outline of what I'll talk about today. I'm going to talk a little bit about the background of the disease, go over just a little bit of biology, and I know that sounds kind of academic, but I'll try to keep it very practical. And then I'm going to talk about symptoms. Okay, I'm getting a little feedback here. There we go. There we go. So, Sally says she can't hear me. I'm going to go over symptoms. The symptoms I'm going to go over today are, I'll talk a little bit about some symptoms that are unique to the greenhouse high tunnel situation. Then I'll talk about symptoms, the kind of symptoms you can see both in the greenhouse and the field. And I'll talk a little bit about pepper symptoms. I'll talk a little bit about diagnosis, but Sally Miller will talk a little bit about that as well. The disease cycle, and then finally leave the hard part, management, to Sally then. My point here was just that the disease was first reported in Michigan in 1909, so it's been known for over 100 years. It now occurs wherever tomatoes are grown. And other names you might see in the literature and in your searches might be Grand Rapids disease, bacterial tomato wilt, stem rot, and bird's eye spot. But those all refer to what we now know as bacterial canker of tomato. All right, so the biology of the organism then. The bacterial canker is caused by bacterium. Clavibacter michiganense, subspecies michiganense, and I may refer to this from time to time as CMM, and you'll see CMM in the abbreviations on the slides. So bacteria then interplants somewhat passively. So if you think about a fungal disease have infection pegs, a lot of them do, which kind of force their way into the cell of the plant, whereas bacteria, their water soaking or wounding is required. And I'll talk a little bit more about that, but that's an important distinction. But leaf wetness is required. CMM then can enter a host by stomates, that is pores or small wounds. So in this slide here, you see the tomato plant, and you can see kind of in sharp relief there, you see the little tiny stomates or plant hairs on the surface of the plant. If any one of these becomes wounded, that becomes a possible infection court for the canker bacterium or any other bacterium for that matter. Also, in the lower portion of this photo, you'll see a picture of a stomate on a plant leaf. And this is another way that the bacterial canker organism can gain entry to the plant. If you think about it, water covering the surface of the plant, that's needed for bacterial infection, but it also needs to be, the water needs to go into the leaf so that the leaf becomes water soaked so that the bacteria can actually go ahead and swim or be forced into that small opening there. So that's why you come out in the morning, you look at the leaf, you see some water soaking, or after a storm you see that water soaking. That's what has to be present. After entering the plant, then the CMM bacterium then can become systemic in the xylem of the plant, so it moves up the plant, and this is important in understanding the disease and in controlling the disease. So if you look at this picture here, then you can see the arrows represent the movement or possible movement of the bacterium in the plant. So when we talk about other bacterial diseases like bacterial spot or bacterial speck, for example, and we can see, we know we're talking about protecting leaves or portions of the plant so it doesn't spread any further. With bacterial canker, then once the bacterium is in the plant, it's in the plant, and there's really nothing you can do about the plant at that point. We want to protect other plants from becoming infected, but that plant, you can put all sorts of pesticides on the surface of that plant, and it's not going to help that individual plant anymore. And you'll notice the arrows go into the fruit here. And in this case here, we see the tomato, it's lopped open so we can see the seeds. That's another important aspect in this disease. Since it is systemic, the bacterium can go right into the seeds. Also in this photograph here, you can see lesions on the outside of the fruit. So if this fruit, there's seed taken out of this fruit, is processed for getting the seed out, it can get bacteria in the seed from the because it is systemic in the plant, and from the outside, from the lesions on the outside of the surface of the fruit. And the fact that this bacterium can be come in on the seeds is an important aspect in controlling this disease. So CMM then does not survive in soil itself, but it survives in crop residue for long periods, especially if left on the surface, on the soil surface. So in this photograph here, you can see the tomato, the crop is on the surface. This is obviously winter. This tomato crop was not plowed in, and it was left on the surface. The bacterium is much more likely to survive in the plant if it's not decayed, if it's on the surface like this. In contrast, if it is plowed in under, as soon as the tomato crop is done, then it starts the decay process, and once the crop residue is decayed, the bacterium is much less likely to survive to the next season. CMM can infect other solanaceous plants, for example, pepper, eggplant, tobacco that is, and several other nightshade species. So this comes into play when you talk about crop rotation, and of course the nightshade species are important as far as weed control. All right, so I want to talk a little bit about symptoms here. I'll start off talking about transplants, go to specialized greenhouse symptoms, and then some generalized symptoms. For example, wilt, marginal chlorosis and necrosis, vascular discoloration, and of course the fruit lesions. So this leaf symptoms here, this photograph is provided by Mary Hausbeck. This is a tray of transplants, and at first glance it looks good. It looks like it's ready to go out of the greenhouse and into the field, but a careful inspection will reveal something's wrong here. But the point here is that these symptoms would be very easy to miss coming in on transplants. But if you look closely here, you can see the etching or lesions on the leaf and on the petiole there of the leaf. And again, once it's inside that seedling, it becomes systemic in that seedling. You can stop it from spreading to other plants, but you can't really do anything about the systemic nature of that bacterium in the plant at that time with any kind of pesticide. All right, so symptoms. These photographs are provided from Sally Miller, provided some of these unique symptoms on greenhouse and high tunnel situation that's a little different, well, quite a bit different than what we might see in the typical field situation. So For example, you might see in a greenhouse situation, you might see these blisters on the leaf here. That'd be very easy to miss, but it's a telltale sign. You also see some marginal necrosis, which is pretty typical, or it will be typical of a field situation. Here's some cell collapse, interveinal cell collapse you might see in a greenhouse type situation. A more advanced situation, you see the cell collapse here, and this doesn't look like a typical field symptom, so you might be aware of this. The stem discoloration can occur in the greenhouse, or it might occur in the field as well. Wilting can occur in the greenhouse like this, or the leaf necrosis. I'd like to move on then to other general symptoms that might occur in the greenhouse or field. And again, there's this wilt symptom that you might see. Remember, if this pathogen is systemic in the plant and it's in the xylem, for example, it can affect the xylem, rot the xylem, and when it does that, the plant has no way to take up water, and thus you might see wilting as is shown here. When I see canker, a lot of times I'll see this symptom along with it, and you see this kind of what I call, it looks like a marginal chlorosis and necrosis that is browning and yellowing along the edge of the leaves. So you can see that here, and this is a real typical of canker, but that kind of marginal chlorosis and necrosis can be typical of other diseases as well. Okay, and I see James Quinn is saying that we saw the more advanced foliar symptoms of wilting in a greenhouse, and that was confirmed with canker in Central Missouri this spring. So I guess some of those greenhouse symptoms were seen in Missouri this past year. When you look at this photograph here, you see that marginal, that what I call a firing symptom. I find that when we'll talk a little bit more about diagnostics. If you're using a diagnostic kit, for example, or you're looking for bacterial streaming, I find that these leaves are a good place to start, because if you cut that open on the edge there, the bacteria really come out of there in great numbers. So that's a good place to start. Also notice in this photograph the proximity of the symptomatic leaves to the stake here. And it is known that the bacterium can survive for periods of time on a stake. So treating that stake and keeping that clean would be one way to stop the disease overwintering from one season to another. And just similar symptoms in the field here. Again, you see that marginal symptom on the leaf. I mentioned that the bacterium is systemic in the plant, and you can see the vascular discoloration. So once that bacterium is going up the plant, it's causing discoloration, and this is part of why the plant wilts and is stunted and may die, because it's almost being rotted from the inside out. This is a typical symptom, but this is more advanced. The entire pith of the plant is mealy and necrotic, and as you can imagine, the plant doesn't survive long after you get that advanced symptom in the stem of the tomato plant. But the bird's eye symptom on the fruit is very typical. I don't always see fruit symptoms, but when I do, this really stands out. So you have what they call it bird's eye, because you see kind of almost like the whites of the eye, kind of ghost-like area around a darker discolored area in the center, so it looks like an eye type symptom. And you see in the background here the firing symptom on the leaves that were found adjacent to the fruit symptoms. This is from the field, and you can see again that ghost-like lesion around the lesion itself, and you see it's almost raised and then sunken again. And of course, obviously, not only does this indicate that the bacterial canker is present, but it also ruins the quality of this fruit. Although this is on the surface only, probably consumers are not going to want to purchase that. I just took this one just like a week ago, this photograph, of symptoms on a tomato in the field. And you can see in the background too, you can see the necrotic leaves, the marginal necrosis, or what I call firing. And this, just a close-up, a closer photograph of that bird's eye symptom. And it, as you can imagine, if you were to run your hand over the surface of this tomato fruit, it's raised. And so this is an excellent way to distinguish this bacterial canker. And what I've done here is shown 3 photographs, one of bacterial canker with that raised bird's eye symptom on the fruit, and then bacterial spot and bacterial speck. In my experience, the symptoms of bacterial spot can vary, and this is kind of a scabby type appearance here, but I never seen them look anything like the bacterial canker symptoms, so they're clearly seem to be much different to me. And then bacterial speck is maybe a little bit raised, and the symptoms might be a little bit smaller, more pinpoint-like. So this is a representation of the 3 most common bacterial diseases and their symptoms on the fruit. Symptoms on pepper. I don't see a lot of symptoms on pepper, but I think it's important to know that it happens on pepper and be able to distinguish that when it does happen. So these symptoms here are raised, kind of rough looking symptoms on the underside of the pepper leaf, and on the top of the leaf some symptoms here, and you see also along the margin of the leaf, kind of almost a little similar to what you'd see on tomatoes. And then the symptoms on fruit look fairly similar to what you'd see on tomato fruit as well. And again, this is going to ruin the marketability of this fruit. Just the upper surface of the leaf, you see how it's raised a little bit. It would be easy to mistake this symptom for other problems. And again, on the underneath, you see the kind of the raised, erumpent lesions on the leaf. So bacterial speck is caused by a xanthomonad, and there's the question here is what organisms cause bacterial spot and speck. There are 4 different xanthomonad species that cause a bacterial spot. It used to be, when I went to school a long time ago, it was Xanthomonas campestris, Pathovar vescatoria. Bacterial speck is caused by a pseudomonad. Bacterial spot has been divided into 4 different species now, kind of a complex situation. If you were to isolate speck, and those folks that do that, it's a pseudomonad. It's a fluorescent pseudomonad on King's bee medium. But like I say, the situation with spot has become somewhat complex in the last couple of years. So I just want to talk a little bit about diagnosis, and maybe this is a discussion that Missouri and other people need to have, is what constitutes a good diagnosis of bacterial canker. Can you look at the symptoms in the field and say that's canker? For an official diagnosis, perhaps you want to go further than that. Perhaps symptoms are not sufficient. So symptoms plus one of the diagnostic test kits, and I know Sally plans to talk about that. And there are a couple of them that are out there. Or symptoms plus bacterial streaming, and I have a photograph here of bacterial streaming. If you had a microscope and a place to do this, it would be possible to be trained in bacterial streaming, and I don't know that we want to talk about that here, but that would be something I'd be glad to help with, and I know Sally could help with training on that. What I like to do in my lab is to have the symptoms of bacterial canker present. I like to test it on a diagnostic test kit, and I like to see streaming, all 3 of those, before I call it bacterial canker. And then finally, there is send it to diagnostic labs, and I think in Missouri that's what you've been doing, is using the a diagnostic lab. And my understanding you don't have one in Missouri now, but will have. And in the vegetable guide, the MX384 is what it's called in Missouri. There's a list of several different diagnostic labs, and also how to send things to a lab, how to send samples to a lab. The disease cycle then, primary inoculum, how does it get into my field is a question I get from growers. Infested crop debris. So if improper rotation, improper fall tillage, if it can certainly come from that debris, and that's kind of the first thing to look for. Volunteer plants, whether from last year's crop or maybe somebody threw cull tomatoes in a field that was going to be used next year. Infected weeds. The bacterium can overwinter on infested weeds. We talked about tools and stakes, how the bacterium can be on those implements. Transplants are very important, whether raised by yourself or someone else. And then all important, seeds. So seeds, the incidence of seed transmission may only be 1 or 2%. So if you think of maybe you have 500 seedlings in a greenhouse that was delivered to you. If you start off in a greenhouse with 500 seeds, maybe only 5 of those are infected, but it can spread in the greenhouse, and by the time you're done raising them after 6 weeks, it can be many more than that, and of course then it can spread in the field as well. But whether in the field or in the greenhouse, splashing water, whether from irrigation or from rain, tools and workers can also spread it. Temperatures that favor this disease are warm temperatures, 75 to 90 degrees. That kind of seems cool by our standards this summer in Indiana, but parts of every day have been in that 75 to 90 degree period, and therefore favorable for bacterial canker. I just have a cartoon here of the disease cycle then. So you have pathogen survival in the seed, crop residue, volunteers, weed hosts, stakes, tools. And you see here the yellow seedling up top here. I think somehow, yeah, so this seedling here, yeah, represents a diseased one. James Quinn is asking, does very hot weather kill it? Last year, after some really hot weather, it seemed to go away in the field. I don't know if it kills it. I think it stops the spread. It may slow it down. My guess is probably there's some still in that plant, but you're going to slow it down. It's interesting you should ask that, James, because I haven't seen a lot of bacterial canker in the field this year, and we've been very warm. We've had temperatures over 100 degrees. But we've also been very dry, and we have had hot weather over 105 degrees. So I would say it slows it down, but doesn't necessarily kill it. And I don't know if maybe Sally will have a comment on that as well. So if you have a seedling like this, and this is once you go to the field, then it can spread by water in the field from a diseased plant here to a healthy plant. And either way, once you have a diseased plant, you have the seeds that are in that tomato, whether it's surviving as culls in the field, and you get volunteers, you saved your seeds, or you purchase or sell seeds, the bacterium can be transmitted in the seeds. Also, the disease cycle can be continued in crop residue. That dead plant can spread it to next year or a couple years down the road, especially if it's not tilled under. The comment is the disease seems to be worse in the first crop when it's during more cool weather. The second planting, when it's drier, seems to be less incident. So certainly wetter weather does spread the disease. It can be too cold for the disease, but it can also be too warm for the disease. Whatever the conditions we've had this year, dry and warm for most of the season, it seems to have been hot enough that it slowed the spread of the disease and the incidence of the disease. At any rate, whether it's on the seeds or in the crop residue, that will complete the cycle then. This is the end of part 1. So my conclusions here: bacterial canker is economically important. It has some unique symptoms, and I'd point especially to the fruit symptoms. The disease can stunt or kill a plant, can reduce yield or fruit quality, and my section of the talk is I'm trying to convince you that it's the disease is best managed when the pathogen and the disease are understood. I've had a lot of good comments. Jaime, if you want to kind of get in here, but I'd be very willing to take more comments at this point, whether the chat box or however, before we move on to the management section.
Thank you, Dan. This is Jaime. Yes, I think we have time for questions. If somebody wants to use the chat box to ask those questions, we have maybe 5 minutes, and then Sally Miller can continue. Thank you.
Sally, I don't know if you wanted to talk about the species of bacterial spot that question was raised.
Well, yes, you're right. There are about now 4 species. The interesting thing for us is that in processing tomatoes, at least, that we found a changeover from the Xanthomonas uvesicatoria and vesicatoria that we used to see 20 years ago to a newer one called Xanthomonas gardneri. And this is different than what they see in Florida, for example, which they see a lot more of something called Xanthomonas perforans. So these species seem to have some differences in the environmental conditions and such that they like. What we don't know is if we're seeing that same gardneri in fresh market tomatoes or peppers, and so we are trying to look at that this summer.
I have 2 comments here, one by Tim Baker. Can buyers who purchase bacterial canker infected tomatoes at a produce auction bring it back to their own operation? Those buyers who also farm, what is the real danger? So I would say any time that you purchase tomatoes that are infected, so I guess what you're asking is not tomato. When you say purchase canker-infected tomatoes, you don't mean transplants. You mean actual produce. Is that right, Tim? Yeah. Okay. So if you bought canker-infected produce and then you took that produce and decided you didn't like it and threw it in the field, then yeah, that would be a possibility. But you'd have to really kind of throw that in the field and then go back into that field and right afterwards or the next year or two, and then that would be the danger. So any produce that you don't want, whether it's from a produce auction or whether it's your own, you want to compost that, get it well away from the production area. I would say that would be a good point. And then James Quinn talks about, we are mystified in which seed source the disease came in on last year, and what can you say about identifying seed sources? That's a huge question, James, and I think the best you can do is to make sure that whoever you're buying the seed or transplant from, the seed has been tested and that it's been grown and inspected along the way, that you inspect it upon delivery. And as far as, I don't know that there's any one seed source or seed company that has had problems or is immune to this problem. And Sally, I don't know if you want to comment on that as well.
Well, yes, I would comment in that I agree with you. This can come up from various different seed companies. Of course, now for tomatoes, there aren't that many independent seed companies anymore, but it does require really good management from the seed company's point of view. But the main point I wanted to make is, first of all, that the major seed companies test seeds for bacterial canker, but they only test a small sample. So there may be a million seeds in a lot, and they can only test a few, right? So they're testing 30,000 seeds out of a million seed lot. So they understand, everybody understands that things can be missed. And there are also technical issues that can cause that bacterial canker, to be missed. So it's not when you get it, if you get certification, you'll see tested and found not to be present. They won't tell you that the seed are guaranteed not to have bacterial canker, because it's not possible. So these are the bacteria can sneak by, because they could be very low levels in the field and the way, or sorry, in the seed lot, and the way that the way that hybrid seed production is gone. Mostly that's done in Asia on very small farms, and it's done. All this pollination is done by hand, so you can imagine that there's a lot of handling of the plants, and so if that bacterial canker is in there, it can move around, and perhaps sometimes it even can be spread without having major symptoms in the field.
Okay, there's some talk about a particular variety, and I think I don't know that it'd be tough to say when something comes in on a variety, unless you can get that seed source. It's pretty hard to say whether it came from a particular variety or not. Uh,
I
Go ahead.
It's been my experience that the seed companies will absolutely deny that they have bacterial canker in their seed lot. So it's a uphill battle to try to get them to admit that, if especially if they tested and they found nothing. So if this is happening again, I'll be talking about seed treatments, and I would say if anybody really wants to use scarlet red, they might want to do a sanitizing seed treatment to help reduce the chance that they might get bacterial canker.
Part of it, I think, what Sally's talking about is the when they test for bacterial canker, the seed companies, it's a destructive sample, so you can't test all. She gave an example of a million seed lot. You can't test all million seeds because you wouldn't have anything to sell then. So their only choice is to test a portion of it, and that's what they do. I see. We got some typing going on. As far as I'm concerned, we can move on. But if there's questions, I'll be glad to. Oh, here we go. Let me just mention. James Quinn brings up greenhouses. I don't see why it wouldn't be the bacterium, the disease wouldn't be associated with greenhouse varieties as well as uh, field varieties. I don't see any reason why that wouldn't happen. Um, but
It has.
okay. In the greenhouse, at least in Indiana, the temptation is to we grow tomatoes year after year, whereas people will do crop rotation in the field. 3 or 4 years in Indiana, we tend to grow tomatoes year after year, and I guess this gets into control, Sally. But if you're going to do that, what I recommend is to take those tomato plants out completely as much as possible, remove them from the greenhouse, and get them well away from the production area for next year. And then I like to see some kind of a landscape cloth over the entire surface of the greenhouse, and that way it's easier to clean and sanitize it between generations. And I think there's still some more discussion, so we'll just. So what I meant in the greenhouse tomato seed. So whether it's a greenhouse tomato or the seed grown in the greenhouse, is that what you mean, James? I wouldn't be surprised at all to see that. Uh,
Thank you, Dan. This is Jaime Piñero. I think I will stop sharing this presentation so that Sally can get hers. Okay, so now Dr. Sally Miller. Thank you. I need to mute
Okay.
my.
Oh, good morning. And thank you, Jaime, and thanks, Dan, for a really excellent uh, introduction and discussion of the biology and diagnosis and symptoms of this disease. Well, I have the title here of Managing Tomato Bacterial Canker. I, like Dan, have included uh, other bacterial diseases, the top three, bacterial spot, speck, and canker, in the management discussion. Really, bacterial diseases in tomatoes are so difficult to manage once they get established, as Dan mentioned. And so integrated program is required, and this is largely a preventative disease program, okay, disease management program. If you wait until you have symptoms like you see in this picture, it's too late for that particular plant. So there is a 7-step program I want to talk about that starts at diagnosis and includes all the potential tactics or methods that ought to be used as appropriately to try to manage not only canker but these other diseases. Now, but diagnosis sometimes gets sort of short shrift because people prefer to just sort of look at symptoms and expect that they can guess it correctly in the field, but it doesn't always happen. So we run a diagnostic lab, as Dan does, and we do a lot of diagnosis of bacterial diseases and use the same kind of tests that he was mentioning. But I also want to mention a couple of the rapid diagnostic tests, mainly because there are a couple issues that I just want to mention for those of you who might want to use these tests. So these field usable diagnostic tests are available only for canker at this point. We'd love to have one for spot as well, but with those 4 species, it makes it kind of hard to develop one. But these also can be used in the lab, but they are very nice because the tests are quick. And if you combine that in the lab with bacterial streaming, as Dan mentioned, it makes it very fast to give an answer. In fact, if the grower is standing there, we can give an answer. These 2 that I want to talk about are the Aodia immunostrip test. This particular one, I couldn't find a picture with the CMM. This one was for Ralstonia, but also a tomato pathogen in southern parts of the U.S. and in tropical areas. We don't have it in the upper Midwest, at least. But anyway, this test for CMM, like all the other immunostrips, is very quick. It just takes about 5 minutes, only has 2 steps. You just grind the sample up in that bag you see there at the top picture, and then you stick the little strip in, and within a minute or 2, you can get a result. You could do this out on the back of a truck. You could do it, you can grind against a rock. You could do anything you like. It's very easy to use. But it does have a problem that's emerged in the last couple of years in that sometimes you get false positives, and what we found particularly problematic is getting false positives when you have very necrotic tissue, like those firing of the foliage that you saw. So as it turns out, and Aodia now has a technical note about this, is that this test for some reason reacts with a couple of very innocuous, omnipresent bacteria in the environment. And so if they happen to be up in those foliar tissue, there's at least one of them is a soil-borne bacterium. So if they happen to get in there, you can detect them. This test, there have to be quite a few of them because this test only detects about 10,000. The detection limit is about 10,000 cells, bacterial cells per milliliter of the sample. So there have to be quite a few of them. So you have to use this Aodia strip. If you're using it in the field, that should be only used in those circumstances as a initial screen, and that should be backed up before any major decisions are made. However, we feel like we can get around this problem by looking for stem lesions. If you find the stem lesions, I would expect it probably works fairly well for fruit. Without necrosis fruit that are just the early lesions, or if you take a stem lesion, you break the stem or slit the stem open and only take some of that discolored tissue from the inside where those environmental bacteria are not likely to be found, and we've found pretty good results that way. So I'm saying you still can use the immunostrip test, just be aware that there sometimes could be problems with false positives, and you can find that technical note on the Aodia website. Now, I also would like to mention a very new test that's just being developed by Envirologics, another company that's in Maine. And this test is more complicated by far than this immunostrip test. But it's also very, very specific, and it gets around the problem of the immunostrips, of cross reaction with other things. It's This one is actually based on a DNA test, whereas the Aodia immunostrip is a serological test, and it is essentially, for those of you who've had PCR tests run by other groups, like by diagnostic labs, this is a type of a PCR test, but it has multiple steps. It still can be used, I wouldn't say out in the hot sun because it takes a while and there are multiple steps, but you can do it inside of a building in a room with just a table. You don't need anything special. Everything's provided. It is expensive, whereas the immunostrips can be as little as $5 a piece. Right now they're saying about $40 per test, which you see on the right there. This again is advertised, we don't have data on this, but it's advertised to be about 100 times more sensitive than the Aodia immunostrips. Now, Aodia is also working on a similar DNA kind of test, a little bit different technology, but they're working on one as well, but apparently, as far as I know, it's not out yet. So these are tests that you may have access to at some point. Just be aware of the pluses and minuses of working with them. Now, we talked about why diagnosis is important, and of course, again, if you don't have an accurate diagnosis, you're not going to put in the appropriate management tactics. An example I always like to use is that at very early stages, bacterial diseases can look similar to fungal diseases, particularly septoria, for example, because you may have spots. You may have spots on the leaves that are pretty nondescript, and if you don't have a hand lens, or if the little pycnidia that septoria makes are not very obvious, it's sometimes difficult to tell, particularly people who don't have a lot of experience. And you can control septoria very easily with fungicides, but bacterial spots not. So you could be not only wasting money if you have bacterial spot and you're putting fungicides on for septoria control, you're also not putting on things that would help with bacterial spot, for example. Now, within the bacterial diseases, it is important to know which one you're dealing with. And for example, one of the reasons that canker and speck survive longer in the soil, always associated with, they're not soil borne, but they're associated, as Dan said, with crop residue for quite a while. Canker also can survive on seeds for a long time. We have naturally infected seed lots that in our laboratory that we isolated about, I'll say 7 or 8 years ago, we still get a low but countable number of canker bacteria off of there. So it definitely can survive. Whereas spot is not only short-lived in soil debris, it also doesn't survive on on seeds as long as canker can. So in a temperate climates, we found in general that the Xanthomonas survives is really dead by the spring following the year of the outbreak. However, the Xanthomonas gardneri, some preliminary results in Canada indicated it might survive past that first winter, and we found also that I have a graduate student working on this, and he also found survival. Of course, we had a particularly mild winter this year, so that may have had a role, but spot generally is considered, in our climate, short-lived in the soil. So we are talking about seeds, and to me, the seeds are the primary point where we should be looking at management of this disease, but it's also the toughest one to deal with because it doesn't always fit within the system that we're using. So this will be more understandable as I move along. But basically, right now, all of the seed treatments that are effective require the seed to be wetted. Okay, so you have to wet the seed with where we'll talk about hot water or dilute Clorox solutions. So if the seeds have been primed, this can cause a problem. And if the seed are pelleted, then you really have a problem because it just destroys all the pelleting. So generally, if a grower, for example, this variety Scarlet Red, if the growers decide they want to treat that seed, they're going to have to ask for it from the seed producer prior to its being pelleted. Then they're going to have to deal with planting non-pelleted seeds or send it back to an organization that does that pelleting. So there are issues about using the currently available seed sanitizing. They're not issues with how well they work. It's just issues with how they fit into the system. So again, check with a seed provider. The seed lots should have been tested for bacterial spot, speck, and canker. Those are the three they always test for. So you should, if you're working with a major seed company, they should do that. If you're saving your own seed, for example, you have heirloom varieties, those should be, I really recommend that those are going to be treated, because especially if you've had any kind of disease problems, but it's not that difficult to do it. And we have growers who are routinely treating their heirloom varieties. If a grower is organic, they actually still can even use the Clorox treatment as long as it's well rinsed, and that's required anyway. So we have a fact sheet that the URL is printed here in Ohio Line, and it shows step by step how you could do the seed treatment with either the dilute Clorox or with hot water. And those are very effective treatments. Normally, if it's a small grower, it doesn't have a lot of seeds, I recommend, even for canker, I recommend the chlorine treatment, just because it's a lot simpler treatment. It doesn't require the precision that you need with a hot water treatment, or the expense, because we usually recommend getting 2 hot water baths, and that's going to be a couple $1,000s. So unless a bunch of growers get together to do their seeds, and we have seen some instances of that, it's kind of better to stick to the chlorine treatment. However, the chlorine treatment potentially could miss bacterial canker that if it's inside the seed, because it's basically a surface treatment, whereas hot water can kill the bacteria inside or under the seed coat. These are some lists of some sanitizing seed treatments, and we've been looking at this for a while, and you can see the whole list there on your left. And one of the issues that again has come up with using these treatments, besides potentially just the incompatibility with the system of pelleting or priming, is that there has been concern for loss of vigor or reduction in germination. And so we have tested that with this particular test of seed vigor. And what we found is that these back down here in the orange box don't affect the vigor of, not in a significant way anyway, compared to the untreated controls. And also those are effective in killing the bacteria. So you have hot water. This is hydrochloric acid, which is used fairly commonly. Also, we tried hot water with sodium hypochlorite or bleach, and then Klingo and Virkon are a couple of disinfectants, and I'm going to talk about those later. So all those are effective, but the two that are are just the Clorox and hot water treatment are on the fact sheet. So we just hear some data we did produced a while ago. It's just looking at the effect of hot water treatment on bacterial canker and spot. And essentially you can see in the picture at the top where the seed were hot water treated, and then where they were not in the lower picture. And this was bacterial canker in this particular instance. So we had a pretty high rate of seed infestation, but you can see there quite a difference when you don't have the disease as to when you do. And that's the same thing it's showing on the graph there. But there are issues, again, as I mentioned, with hot water treatment, and this is, I think this, hot water treatment and Clorox treatment are old treatments. We first started working with them 20 years ago, and nobody even knows who initially developed these, as far as I know. We weren't able to find it. It's just sort of floating around in a lot of different recommendations. So, but what you can find is that sometimes, as you see that blue line on the left in the graph, is that occasionally you'll see that the hot water treatment can reduce germination or delay it. And again, in the end, by the day 6, everything was germinated, but one of these was delayed. And sometimes this happens if the seed lots are old or poor quality. And in such cases, the vigor may be reduced as well. Also, we only recommend that these seed treatments done immediately prior to planting, so you don't have any issue of seed longevity. And again, we mentioned about the other seed treatments. Now this one on the first slide, I put a parentheses around it because, of course, in any IPM program, you want to use resistant varieties. But unfortunately, there really aren't any. For these particular bacterial diseases, there's a little bit of resistance for bacterial speck in some processing tomato lines, but in general, you don't have good resistance to bacterial diseases, particularly not spot or canker. And that's not for lack of trying. These are just very difficult to develop. There are varieties that vary in what the seed companies might call tolerance, which is really kind of a partial resistance, and that basically can hold up some varieties better than others. But as far as I know, you won't find that in the seed catalogs. It's just kind of like people who have a lot of experience with these varieties kind of get a feel for which ones can hold up better than others. Okay, so the next step then is to use pathogen-free transplants. And as you can see, this is actually a quite typical large greenhouse operation, and the small ones, except for the size of the structure, can be very similar in that those plants, those seedlings are jammed together very closely. You might have as many as 300 and over 300 seedlings in a single tray. For processing tomatoes, usually fewer where you have fresh market tomatoes. But that plus the overhead irrigation that's typical in these, whether it's by automated sprinklers overhead or if it's just coming in with a hose, basically you have a lot of seedlings touching each other in close proximity, air movement being a little slow because they're also crowded in there, warm temperatures, and then water. So essentially, it's an ideal environment for the bacteria to spread. So the other issue is that we saw symptoms of in the greenhouse in a flat, but those are a little bit developed. Sometimes the symptoms are so unnoticeable or so small that they're never seen when the plants go out, or the bacteria may just be surviving on the surface of the plants and not actually having gone in and caused the problem at that point. So the levels that are required to get an epidemic going can be developed in a greenhouse without ever seeing much in the way of noticeable or obvious symptoms. I just want to show a slide here of some work done by one of my graduate students, Shulan Shu. And what she did was she engineered the CMM, one strain, with a gene that causes the bacterium, as long as it's alive, to glow in the dark. So basically, what we can do with that as a tool is to look at how this bacterium infects tomato seedlings and plants. So what you see here is some of the early work that she did, and she inoculated this glowing bacterium onto seeds, and you can see if the first day there in the picture, the brighter red the color, the more bacteria there. But you can see that as the seed germinates, the bacteria stay there, and then here at 5 days on the right-hand side, you can see that there are bacteria still stuck on the seed coat up at the top, but they're also on the hypocotyl as well. So this would be in a real-life situation already above the soil or the planting mix line. What you don't see here, because the sensitivity is not quite enough, is that they're also colonizing the roots. So they are in a position then, up above the ground, to infect, for bacteria to move around from one plant to the next if they are in that seed lot. So what should growers be doing? The first, they should, despite the fact that we said that they're hard to see, but they should be scouted regularly for bacterial canker, spot, speck, or any other problems in the field. These plants who have the symptoms should be destroyed. Now, again, they should be confirmed by a diagnostic lab or a kit, because there are some things that actually look like spot or speck or canker. It's really hard to tell at this point. but are not a disease. For example, when a lot of times these growers are holding the tomatoes, they withhold phosphorus and other nutrients, and a phosphorus deficiency or other nutrient deficiencies can result in these, especially as plant seedlings get a little older, these sort of necrotic spots on the lower leaves, and we need to test those to make sure. Normally, we would just quickly do that in the lab with a bacterial streaming test, but you can use the other tests as well. Now, if this is confirmed to be particularly canker or spot or speck, the flats that have those symptoms should be destroyed, and then a certain number of surrounding flats should also be destroyed. It depends on how much is seen, how many spots are there, and how much risk that the greenhouse grower or the customers are willing to take. So it's basically at least the flats that are adjacent should be removed, and probably several layers of adjacent flats should be removed and destroyed. And again, they need to be composted far away. They shouldn't just be thrown outside, especially not near a water source. Then the area needs to be sanitized where the flats were removed, and also all the tools that might be used should also be sanitized. And then again, additional scouting needs to be kind of ramped up in order to, particularly in that area, to see if it is spreading over the period of time until those plants get outside. I want to talk a little bit about sanitation, and because sanitation in both the seedling production phase and the field production, whether it's in the open field or greenhouse or high tunnel, is really critical, particularly with bacterial canker, but with all bacterial diseases. And again, because we can't very well control this disease, any of these diseases after they've gotten established, it's really critical to prevent them. And sanitation, unfortunately, requires a lot of diligence on the part of the greenhouse operators or the growers. And people get busy, and it's sometimes difficult to keep up, but once a problem has happened in an operation, people are more willing, because of the losses they can get, that can come about, they may be more willing to try to look at these practices. So again, the first step is that the operation for seedling, I'm talking about seedling production now, needs to be as clean as possible. As Dan mentioned, floors, if they can't be concrete because of the expense, I also agree that some sort of ground cover, a nursery cloth, whatever, will help to keep things clean and for sanitation at the end. So I think that that's really critical. And of course, that the benches should be raised some way so that none of the flats are sitting on the floor, on the ground. The coal piles should be, if there are coal piles, they should be very far away. Again, composting is better. They should be composted right away. And that if you are composting, it shouldn't just be stacking them up on a big pile. It should actually be turned on a regular basis so you get that rapid decomposition. Weeds are really critical, particularly solanaceous weeds, but even other weeds can reduce the air movement if there are enough of them, and actually cause problems, not just with bacterial disease, but with other things. There should be no volunteer tomatoes or peppers. That picture on the right is one I took in a greenhouse where a volunteer tomato was allowed to come up, and if you look at the lower part of that leaf there, it's got bacterial canker symptoms on the leaf. So if you can imagine, you had a bad outbreak of bacterial canker in a greenhouse, and some of those seeds fell to the ground and were allowed to come back up, they certainly could transmit the disease. Again, no plant refuse on the floors. That needs to be all cleaned up carefully. And we've been working with a lot of greenhouse growers, small and large, and we were surprised to find a lot of small greenhouses that let their dogs or cats in the greenhouse. I don't think that's a very good idea at all. They can move things around just mechanically if they are touching plants. Okay, so I'm going to talk a little bit more about sanitizers, but one thing is that tomatoes are usually germinated in a germination chamber, so that, and we find growers tend not to sanitize that very often. We'd like to see it done after every seed lot that goes in there, but that's probably often not practical, but that should be done as much as possible. The house needs to be thoroughly cleaned and sanitized. That includes the benches, equipment, tools after every crop. Workers need to have clean clothing, and it's best if they have shoes they only wear in that particular greenhouse. Foot baths and should be at the doorways if with active disinfectant, and that is a big issue because a lot of times people put disinfectants in the foot baths and then don't think about it again for weeks, and of course it's no longer active. And obviously only use new or sanitized plug trays as well as planting mix. This should be new. Well, these are some of the common disinfectants. They're in groups: alcohols, halogens, like the chlorine, bleach, peroxides, and quaternary ammonium compounds. And we've tested a bunch of these, including the ones in blue, and these all were effective against bacterial canker and the other well, we haven't tested the other bacterial diseases, but they have been effective against canker. Doesn't mean the other ones are not. These are just the ones that we've tested. So how do you get the most out of these disinfectants? Sometimes I'll call them sanitizers, the same thing. First of all, growers need to read the labels and make sure that they follow the instructions. Again, the biggest issue is I think, is growers letting it go too long before they replace it, so that they're kind of using inactive materials when it should be, and they kind of get a false sense of security. Some of the sanitizers, disinfectants are corrosive, like chlorine, bleach, for example. Vercon's also corrosive, so very thorough rinsing for tools and surfaces is necessary. Of course, organic matter destroys the activity of these sensitizers. That's how they react, particularly the peroxides and chlorine bleach. So again, that disinfectant needs to be replaced. Okay, so the question here is there any way to sanitize wood frames of benches? One grower said if they are wrapped with plastic, doesn't that make it okay? Well, if they are wrapped with plastic, it makes it easier, I suppose, because you're just kind of sanitizing the outside, the plastic, and getting it clean. If they're wooden, I think even these sanitizers can work quite well on wood. For example, I think a quaternary ammonium compound might work. You just have to look at the labels. I think it's possible to do it. What I didn't write down here, and I should have, is that all of this, the sanitation should be done as a second thing that comes after a very thorough washing with like a hot soapy solution and rinsing. And that will take care of a lot of the problem as well. So you really not only relying on the sanitizer. Okay. Now, if we have seen growers who are using chlorine, and we see this pretty often, and they're kind of sort of guessing when the sanitizer is active, but chlorine is really kind of easy to check with there are meters that you can buy. This is an oxidation reduction potential meter. It will help you determine the parts per million. Also, even swimming pool kits can be used. So it's not very expensive to test, and the testing should be done, particularly if you're using something like chlorine for sanitation. Okay, so I want to talk about two things in this slide. One is minimizing the moisture, and we're calling this dry growing, and this is kind of a term that growers have used. And there's not a lot of science behind this. This is kind of experience by different growers that, those growers who are very sparing of the water for whatever reason, maybe not so the plants don't grow too fast, or they tend to have fewer problems with bacterial diseases. And that is understandable, because we have talked all this whole hour this morning about how this disease requires water to move around and to infect the plants. So if these plants are kept dry enough so that the water on the moisture on the plants, the seedlings, dries out quickly, that microclimate is drier than if you've got those plants saturated, we tend to see fewer problems. So you'll find that, even sometimes we'll find that greenhouse transplant producers, may have the same lot of seed, and one grower will have a lot bigger problems than another grower. Now, it's possible that's because, they have the luck of the draw with the seeds, but on the other hand, it's possible too that there are differences in the way growers handle that. So growers should, of course, the first instinct is to give them plenty of water so they don't dry out. and often the flats around the edges will dry out quicker. But this is something that growers ought to have in mind or producing transplants to try to keep them, to just give the plants enough moisture to get them healthy, growing in a good, healthy way, but not to overwater. Now, the other issue is using what type of source water, and there are all kinds of water in a greenhouse, maybe recirculated water, and in fact, the EPA is coming out with more and more strict rules on water runoff from greenhouses. So a lot of greenhouses, at least the production ones, who use a lot of water are recirculating water. So if water is recirculated, that's an obvious that it needs to be treated. And there are lots of different ways to treat it with chlorine or chlorine dioxide, chlorine gas, ozone. There are various methods that are used. They're all expensive, but they're usually effective as long as they're working properly. If it's not recirculated water, if it comes from a well, it's or city water particularly, which has already been chlorine treated, it's usually not a problem. If it's surface water from the farm that comes from a pond or stream, we do know that bacterial canker can survive in water, probably bacterial spot as well for some period of time. So it's really important if surface water is being used that it's sanitized by some method, effective method. Okay, so then I think this is about the last point. The 2 points I want to make is that a lot of times growers will try out some new varieties, and they'll put them in the same greenhouse as the ones that maybe come from the major producers, and that is one way of introducing this disease or one of these 3 diseases into the greenhouse. So we really recommend that if those are, come from an oddball source or you've saved them yourself or whatever, that they really should be sanitized or they should be put in a separate greenhouse. The other thing I want to mention is application of crop protectants, and this has been recommended by Dr. Mary Hausbeck for a long time, is to apply 1 or 2 copper treatments that may or may not contain mancozeb. Mancozeb we usually recommend. And then also an application of agarstrep, which is an antibiotic, streptomycin, before the plants leave the greenhouse. Now, agarstrep's only allowed to be used in the greenhouse, and so that's the only opportunity to use it. It's a good idea to use it because it's kind of a different mode of action, certainly, than copper. So 1 or 2 copper treatments and 1 application. This is to kill the bacteria that are on the surface. And again, if the bacteria have already gotten inside the plant, they're not going to be killed by this, but it's to help reduce any bacterial spread during the greenhouse propagation phase. I want to mention a couple points about copper because copper is primarily what's used for bacterial disease management. And again, I think once you have a kind of raging epidemic out in the field, it's just wasting time and money to put copper on. But early on in the season or in the greenhouse, I think there's the value in doing that. But there are some interesting points about copper. And first is that the residual drops really quickly on the leaves. There was some work in Florida that showed under those outside, now these are both outside conditions, there is very little detectable residue 5 days after application. And similar results were found in Ontario. Again, this is just a report that we were given. A lot of this stuff is not in the literature. So that's why growers in Florida are painting their tomatoes blue almost with copper products every 5 days at least because how quickly that residue drops. The other thing I want to mention is that the activity is affected by the pH. So the lower the pH, the more active are the copper ions. But if the pH is less than 6.5, you can burn the plants. So I think a lot of growers never check the pH. And I think it's really critical that they start doing that because you could be wasting, again, time and effort and also not getting control if the pH is wrong. Or you can run the risk of burning the plants. Again, it's a protectant, so it has to be applied before the bacteria get inside the plant. And then also we prefer, again, that it's applied before it rains, again, because it's a preventative, but it has to be done in time for the plants to dry. What a lot of times will happen is that the copper doesn't get put on and then it rains and then they can't get in the field because it's too muddy and then by then the bacteria really go into town, so to speak. Okay, so let's talk about the production in the field. Again, a lot of high tunnel production in Ohio and other Midwestern states in the last 10 years or so. A little bit different situation in the field in high tunnels and greenhouses than you have in this case a fresh market. And of course, in Ohio and Indiana, we still have quite a significant acreage of processing tomatoes. So these production systems, the diseases you get are a little different. You get canker everywhere, whether it's in the greenhouse, in the open field, high tunnel, or the greenhouse. So it just it doesn't matter. But as for spot and speck, we rarely see that in a high tunnel or a greenhouse because there isn't overhead watering under these conditions. Even though in the seedling production with overhead water, you can move those things around. But in the high tunnels or greenhouses, nobody is overhead watering that I know of. And so you don't have the moisture in there unless there's a big storm and it comes in on the sides of a high tunnel. It just don't see very much spot or speck underneath a high tunnel because it's a big rain shelter. So one of the practices that moving on to another practices in the field is again to choose the best site and to practice crop rotation. And again, with a lot of growers, this is difficult to do because they like to put their tomatoes on the best possible land that they have, and maybe that's limited. But it should be well drained, have good air movement to dry those plants out as quickly as possible after a heavy dew or a rain event. Should be sunny because tomatoes like sunny condition. We found usually that not only bacterial disease, but a lot of other diseases tend to be a little bit worse with the closer that you are to woods, just because the air movement is blocked a bit. And then, of course, weeds need to be under control, particularly the solanaceous weeds. And then rotation. For bacterial spot, at a minimum, 2 years. So at least 1 year without a solanaceous crop. A canker speck, I should have taken out brown spot. That's a pepper problem. That should be at least 3 years, preferably longer. If you're growing organically, you have to do these long rotations anyway. But this takes as long as it takes to break down all of the residue. That's how much time it needs to get make sure you don't have canker in the system. Okay, some other cultural practices, or some cultural practices that I'll spend a little bit of time on now are maintaining reduced stress growing conditions, well-drained soil, not overdoing the nitrogen. Again, especially inexperienced growers, they tend to put in too much nitrogen, get those plants really looking lovely, but for one thing, they don't produce fruit very well under those conditions, and a lot of diseases tend to be worse when the plants are off balance because of too much nitrogen. Again, regular irrigation, drip irrigation, of course, is the best, so you don't wet those plants. And then also the work on improving the soil by improving its organic matter content where that's depleted, and that includes cover crops or compost. These are all well defined for organic growers, and certainly conventional growers can use these practices. New or sanitized stakes or strings, etc. Again, not to introduce these organisms, particularly canker, into the field. Avoid moving among the handling plants when they're wet. And of course, people like to do their harvesting early on in the daytime where there might still be a lot of dew because it's cooler then. But it is the potential for moving bacterial diseases just by brushing them from plant to plant. Sanitizing equipment, hands, boots, tools, etc. between fields would be extremely helpful, but we find in the surveys that we've done that growers almost never do that. It's hard to remember to do it. I would think that at least if it's really a big problem, that they would start doing it. The questions come up, growers express concern about moving from their tomato fields to the adjacent field. Well, yes, that's correct. And again, we recommend that that essentially all that equipment be power washed and then a disinfectant sprayed on it, if possible, before they move into that field. That is a potential, I think, um, they actually have to get some tissue, plant residue in there. So I guess that could happen if soil is moved. I'll show you a photo of, roots actually can have bacterial canker in them. They probably don't survive as long. So I would say that if they can do the one thing is to, clean the equipment regularly and then always leave those fields last, especially if they have a disease problem. Okay, again, pruning and other crop work for in the high tunnels, greenhouses, and fresh market steak tomatoes. One of the things we've noticed is sometimes the growers get behind and they remove suckers by hand. You can do a lot of work by hand and not have to use tools because tools are really spreading. And if you have a lot of practice, you can snap off the suckers and you don't have to use a tool and you don't even touch the plant. But you can see on this picture, there's a big, giant, gaping wound there, and that's a perfect spot for bacteria to get in. So again, just try to remove the suckers when they're small. And then if there are pruning tools, there are some tools available that I can provide information if anybody is interested that actually should deliver, they're expensive, but they deliver disinfectant right to the cutting surface every time you squeeze the clippers. And these are great. They work really well. But again, they are a little bit of trouble because you have to fill them up with disinfectant every once in a while. And also they're expensive. But if don't want to do that, you can dip the pruning tools into a disinfectant after every plant. Again, we're finding practically a lot of growers don't want to do that, but at least at the minimum, at the end of every row to try to contain a problem, particularly canker, again, because it's systemic in plants and you can easily move it. So if bacterial canker is identified in a high tunnel or a greenhouse, those plants, as Dan mentioned, need to be removed. Now, this is a very extreme case in this picture here, and eventually all the plants had to be removed because this came out of the greenhouse with these grafted plants that were probably a lot of them infected. So they end up having to get rid of everything. But essentially, those plants should be placed when a plant is found that has canker, it should be taken out of the high tunnel or the greenhouse in a plastic bag so it doesn't touch any other plants. And also several plants on either side. Now we have some large greenhouse growers that will take out 10 plants on each side of an infected plant. Others won't take out nearly that many. It just depends on the experience of these particular growers. So again, if you have it in a high tunnel, that plant should be taken out, but without touching any other plants, and then cleaning up your hands. The growers should need to clean up themselves, their hands and clothing afterwards. Then again, intensifying the scouting in that case, doing a better job with sanitation, quarantine those effective rows, and work in those rows last. So essentially, it's just using sort of common sense approaches. In the field, it's kind of be pretty unlikely that people will start taking plants out, but they can, especially if it's a small growers. I would recommend they do that. But again, to be very careful not to touch the plant, like put it in a plastic bag and move out without touching any other plants. In the end of the season, here's a picture on the right again with that glow-in-the-dark bacterium, just so you can see how the bacterium moving up from an inoculation point at the bottom of the plant, the lower picture up into the plant, the leaves and the petiole stems. But also I wanted to show you the top one because it just shows you how many, there are a lot of bacteria in the roots as well. So it's really important to plow down those plants and disc them to speed the breakdown of residue. As Dan said, if they're laying up on the top, it survives longer. They need to be in the ground, in the soil, so that the microorganisms in the soil break down that tissue, the residue. And then, again, get rid of all the plastic, the stakes, the strings, etc., and sanitize anything that's going to be reused. And also, I should have said, but to clean the equipment afterwards, especially if you're in a field that's got canker or spot or speck, particularly canker. In the high tunnels, we have some similar issues, just getting rid of that plant material that's there, cleaning the pots and clips and stakes, etc., whatever is used, and then to sanitize the structure. Now, the last thing I want to talk about is crop protectants. And again, we are very limited in what we have in terms of working, how well it works on bacteria. You have to do it very early. We recommend starting a copper application 7 days after transplanting, within 7 days. We do include mancozeb with the copper sprays. Again, it's supposed to be a little bit helpful, increasing the copper ion activity. We keep those antibacterial treatments early in the season to try to keep those populations low and through fruit set at least, and then apply the copper when it is applied on a 5 to 7 day schedule. We also have some success, particularly with bacterial spotting, using Actigard. And Actigard is a plant activator. It helps the plant to be more resistant, and that can be very helpful as well. So that's all I have to talk about specifically. Before I talk about this brochure, there's a question: would working the field twice help the breakdown process more? I would think so, yes. Again, I guess it depends on the soil type and the attributes of that particular field, but I think that whatever you can do to chop up that tissue is going to help it break down more. Okay, so finally, I just want to mention this particular brochure you can download for free. It's available in English and Spanish, and it has a lot of this information that you've heard about today that can be handed out to growers. So I'd just like to thank Lincoln University with Dr. Pinero and Andrew Erb and Jameer Brown for getting all this set up, and those folks that work with me, and of course Dan Egel. I guess that's it.
Thank you, Sally. So basically, it's going to be available at the Lincoln University IPM program website, which is very long, as you can see. So it will be easier if you would like to just type lincolnu.edu. In the search box, you will find them. You can type IPM, and then you will see the website. Once it's made available, maybe in a week, I don't know exactly when, then you can access the PDF of the presentations as well as the PDF or the recorded webinar. Also, I want to try to find out how I can post this in eExtension, because with that I don't have experience, but I will find out how can I post the webinar in eExtension for everybody to see. Any questions? Susan Jaster to Sally Miller. Where can they find the test kit? Can you hear me, Sally?
Sorry, I was looking at the questions. Yeah, the test kits I mentioned, the Aodia immunostrips are available at aodia.com. They could just be ordered online. And then I have to say the other one is, I looked at their website last night. I'm not sure they're actually selling them yet, but they may be. So I think it's just the best to go to envirologics.com. I'll write that down. At envirologics.com if they're interested in that one. Someone also asked about rain barrel water being problematic. I would say probably not. I don't think it would be a problem to use rain barrel water. Dan, what do you think? I don't know where. Yeah, I can't hear Dan, but. Oh, I agree. Okay.
Thanks again to Dan Egel and Sally Miller for their support and sharing their knowledge.
We're still here. If there are questions or other interactions, we'll still be around.
Very good. Thank you.